There are still many things we do not understand about the human genome. The first human genome sequence came out back in 2002, 2003, and it was sort of presented like everything was solved. But in this new version of the human genome, we realize that there are many unsolved mysteries. We can really start to understand the dark matter of the genome, and I think it holds so many answers to questions about human health, human fertility, that we have not appreciated before sort of this moment in time where we are now. Before 2022, there were 5 chromosomes where we never had the sequence of the short arms before. Those 5 chromosomes are called acrocentric chromosomes, and they have a special shape. They have a short arm and a long arm. So what a Robertsonian chromosome is when 2 of those long arms fuse together from 2 different chromosomes, and the short arms are lost. And at that point, instead of having 46 chromosomes, a genome will have 45 chromosomes. Sometimes that doesn't work very well, and that is why individuals with the Robertsonian event often have infertility or subfertility. And so what we found was that there were certain regions, certain very specific regions, these repetitive sequences that are shared across multiple chromosomes called SST1. They're sometimes referred to as junk DNA. They're sometimes referred to as dark matter, and that was very surprising. We never knew that it was actually the same sequences on different chromosomes that could come together and make those fusion events. So Robertsonian chromosomes are not that rare in terms of the human population. About 1 in 800 individuals has one of these chromosomes. These chromosomes are relatively common, but most people won't know they have one unless they have a fertility issue, and then they go to the doctor and have certain tests done. A lot of cases of Down syndrome are associated with maternal aging, but there are cases of Down syndrome that are not associated with maternal aging. And in many cases, the underlying cause in that instance is one of these Robertsonian fusions. It's a really exciting time right now because of all of the long-read sequencing technologies that are available. The long-read sequencing is super important for these really repetitive regions because it allows us to resolve them and assemble them in ways that were not previously possible. What's been really satisfying in the last few years is seeing all the advancements that we can make based on these genome resources, based on the long-read sequencing that is happening. And I think this is just one example of the type of advances that we're going to see as we move forward, as we start to apply these new technologies broadly, not only across the human genome, but across the tree of life.